too much of a good thing. In the world of disease, some mechanisms pathogens employ leave scientists dumbfounded. Certain pathogens have a unique ability to hijack normal functions within our body and turn them against us. The bacterium behind typhoid fever, Salmonella typhi, remains one of these elusive pathogens. Rampant in developing countries due to poor sanitation, typhoid fever infects anywhere from 11 to 20 million people every year, according to the World Health Organization. In a new study, scientists uncovered the mechanism behind the inflammation seen during the disease. The scientists discovered that a gene inducing an inflammatory pathway occurs in high concentration in S. typhi, resulting in hyperinflammation. Pathogens can induce hyperinflammation through targeting a pathway in the body called the inflammasome. The pathway activates cytokines, chemical signals excreted by cells, which promote inflammation. They do this through activating an enzyme called caspase-1. Caspase-1 becomes activated when the cell detects an antigen. This enzyme signals a cytokine interleukin-1 beta to mature into its pro-inflammatory form, while also promoting the cell to undergo pyroptosis. Different from the normal form of programmed cell death, apoptosis, pyroptosis instead causes programmed cell death through inflammation. The cell, instead of shrinking, balloons in size until it dies. A group of scientists from the Molecular Infectious Disease Research Center of Changgung Memorial Hospital focused on the mechanism causing hyperinflammation during typhoid fever. In a paper published by Microbial Pathogenesis, the team looked at mice to understand which form of cell death takes place, as pyroptosis seemed to dictate the inflammatory nature of the disease. A small amount of antigen induced apoptosis, while a large amount of antigen present induced pyroptosis. The scientists concluded that antigen concentration must determine pathogenesis, but what antigen had the high concentration causing increased activation of the inflammasome during typhoid fever? The scientists then looked to how S. typhi cycles throughout the body. How they cycle through the body can have a large impact on the mechanisms the pathogen employs to reach the disease state. Typhoid fever has an incubation phase where the bacteria waits and multiplies undetected by the host immune system. It accomplishes this through an outer coating called the VI capsule. The capsule acts as a cloak covering important structural features on the surface from immune cells. These immune cells, such as macrophages, act as watchdogs, attacking invaders, while also alerting other cells through complex chemical signaling. With the VI capsule on, macrophages have no way of detecting the typhoid cells when they circulate the bloodstream. The bacteria then travels to the gallbladder, where it undergoes a series of fast replications, growing in number. Scientists wondered if the presence of VI initiated the hyperinflammation phase of the disease life cycle, acting as an antigen. They decided to grow two versions of S. typhi in the lab, one that lacked VI and one that had it. They found that both versions of the bacterium expresses the same amount of pro-inflammatory chemical signals that begin the inflammasome pathway. They concluded that VI did not activate the inflammasome, or they would have seen significant decreases in inflammasome activity for the VI negative type. If VI did not, then what did? During the test, the scientists noticed that while VI concentration decreased, SPI1, a set of genes found in the bacteria, expression increased. Did they find the elusive antigen activating the inflammasome? SPI1 genes produced the antigen that in high concentration triggered the start of the inflammasome. The scientists concluded that while in the gallbladder, the typhoid bacteria hyperexpresses the SPI1 genes. Coupled with the rapid replication, this causes a large concentration of SPI1 in the intestinal tract when the bacterium migrates there. The inflammasome then overtakes the intestinal tract and completes the mechanism behind the strong inflammation period of the disease, resulting in systemic infection and tissue damage. Our understanding of how mutations may alter the severity of disease depends on our understanding of the mechanism behind the pathogenesis. With this knowledge of how typhoid fever progresses, scientists in the future can develop new vaccines, better treatment, and determine other mechanisms of bacteria. Overall, this study reminds us that pathogens can wreak havoc by using our normal cell functions against us.